evening to the participants. So, welcome to the day 5 session on molecular talking in internship and certificate course in AI and ML approaches of drug and vaccine design. So, before stepping it to the today's topic session, can we recap the last session topics? So, what exactly did we discuss in the last session? Yeah, discuss with the last session topics. You forgot, isn't it? You want me to show the hint? Now you remember what exactly did we discuss? Yeah, come with your answers. What exactly did we discuss? Yes, we started very good. Initiated from Tania that we discussed on LADMET. What is this called? Pharmacokinetic properties. Okay, liberation, absorption, distribution, metabolism, excretion, and toxicity. Then what else did we discuss? Drug databases. So, what are the different drug databases we have discussed? So, we discussed from Drug Bank, CHEMBL, CHEBI, NCBI PubChem. Any other I missed? So, these are the KEGG drug. Okay, Kyoto Encyclopedia of Genes and Genomes. There also we find the drug database exclusively. Okay, we have plenty of databases, but this here you can find large data. So tomorrow we'll be discussing on virtual screening, whereas in virtual screening we'll be seeing even synthetic molecule database like Zinc 15, etc. Even that can be used as a drug database. Okay, then what else did we discuss? Very limited answers from the participants. So why can't you discuss? Especially, I don't find any kind of responses from SVC students. What happened to you? I think you also studied a similar kind of a subjects, but your interaction is very no interaction at all. Very good. Once again from Tania that we discussed on drug likeness filters. Especially we discussed on Lipinski rule of five. Okay. Can I ask you a few questions? Then I think I can anticipate answers from everyone. So, what are the different drug likeness filters we have discussed other than Lipinski? This is my first question. The next is how about Lipinski's rule of five? So, what are the properties, physicochemical properties of the drug present in Lipinski's rule of five? So, I asked two questions. Mention me the types of drug likeness filters. The next is mention the physicochemical properties of the drugs or the compounds. Lipinski rule of five. Am I audible to the participants? Molecular weight less than 500. Very good. So one of the property in Lipinski rule of five. How about the other properties? Even someone can unmute and answer so that we can minimize the time of interaction. Today we'll be discussing on molecular docking. A lot of points on I have to show, and also I have to explain you theoretical concepts. Hydrogen bond donor less than 5. Very good. Hydrogen bond acceptor should be less than 10. And then one thing is missing. Yes, very good. Log P. Partition coefficient should be less than 5. Okay, so whatever mentioned like molecular weight less than or equals 500, hydrogen bond donors less than or equals 5, and similarly acceptors less than or equals 10, log P less than or equals 5. Okay, so these are something about polar surface area is won't include in Lipinski rule of 5, but it includes in other drug likeness filters. Okay, I asked you one more question, isn't it? What are the different drug likeness filters other than Lipinski rule of 5? At least one can you can mention any one. I can show you the remaining thing. Remember any kind of drug likeness filter other than Lipinski rule of 5? So for this, you have to go to the Swiss ADME tool. And what are the, so you want me to answer? Mugi, Weber, then Mugi, Weber. So these are the thing majorly we discussed, isn't it? Mugi, Weber, etc. Okay, then what are the different tools we have discussed in Swiss website? In Swiss website, we have seen Swiss ADME. Like Swiss ADME, what are the other tools we have discussed other than Swiss ADME? Swiss Param, very good. Like Swiss Param, you want me to show once again? We're also going to work on Swiss Doc. Okay, Swiss Doc. If we have a time, if it's simply type Swiss ADME, directly take you to that website, but you can also see the other options present. Like Swiss doc, I didn't explain you, but today I'm going for docking, isn't it? If you click the Swiss doc, we get to input the smiles format of a ligand and then input a PDB format, PDB ID of a protein. So just these two inputs we have to feed and search space. So we can give the box center box size. So more about the box center size we'll be discussing during the theoretical presentations. And then we can see these kind of interactions in the Swiss doc. Simply uploading two input files. One is ligand, the other is the receptor. Okay, and feeding the grid size. This is actually a grid size. So after fixing a grid size, we obviously will be receiving an output in email. And of course, there's an option. This is not a mandatory. So it takes some time. Okay, so better feed a mail and feed a kind of an project name for this. So docking with attracting cavities option is available. Docking with Autodoc Vino is also available in Swiss Doc. Okay, these are recent implementations. That is docking with Autodoc Vino. Okay, this is an online tool. Now let me go ahead with anything else did we miss other than yes, very good. So we discussed on Swiss target prediction, and then we have gone with the Swiss bioester, and then Swiss similarity. Okay, these are the things we have discussed on yesterday. So in everything we have used. So I can ask you one more question. What is the 1D format of chemical compound? What is the 1D format of chemical compound? The next question is, what are the possible 3D formats of chemical compound? Very good. The answer is canonical smiles as well as isomeric smiles. There are two types of smiles format. If smiles is a 1D format, how about the 3D formats of chemical compounds? So in what formats you can find the 3D formats? Yes, very good. MOL. Like MOL, give me some few more answers. Because this is very, very important, isn't it? If you go for talking, one should know like how to download the molecule if you want to change the format, even in Autodoc, So if you have a ligand and receptor in PDBQT, there is a format called PDBQT. If only if you have in PDBQT, then only we can able to dock in Autodoc. There are many versions of Autodoc. I'll be explaining you about when we go with presentation. Very good. MOL PDB. Then anything else? Only one participant is interacting. Oh, how about the others? Okay. Dot MOL2 dot STF. And then did I miss anything? Yes. MOL2 STF.pdb she has already mentioned. And now just I came with one more format, isn't it? PDBQT, which is present in Autodoc. Good. So these are the most popularly used formats, okay, for chemical compounds. These are 3D structure formats. Okay, now let me go ahead with the today's presentation. Uh, so just learn through the videos, okay? So the interaction is gradually reduced. I anticipate this kind of interaction should be increased in the subsequent sessions. I thank all the participants who, because I got a good feedback from your side, but still, okay, this interaction I found we are lacking. So still we can improve by watching the videos or even practicing whatever I'm going to teach on today's session. Okay, let me go ahead with the molecular talking. So we today will be discussing on molecular talking, ligand protein talking softwares, protein talking softwares, theory of molecular talking, scoring methods, hands-on experience in Autotalk. What is molecular talking? We have already discussed it is the computational procedure to predict the conformation. So conformation also called as pose, that is a ligand binding with a protein can bind with the different poses, okay, because a protein is a macromolecule. You find so many binding sites, and a ligand can dock in multiple binding sites. So in this computational procedure, we predict the conformation of a protein ligand complex. That is, after binding a ligand in the protein, we call that as a complex protein file. And if you do the talking using Autotalk, finally we save this protein ligand complex file as .pdb. Okay, so to check the interaction patterns. And most important thing in molecular talking is like how a freeware is distinguishes with a commercial software that distinguishes from search algorithm and scoring function used in the tool. Okay, search algorithm and scoring function. This is most important in this molecular docking software tools. And if you see this molecular docking, what is the concept? We have to use a small molecule called ligand. So the ligand is usually a drug, and the target is usually a receptor. We try to check the binding patterns. Okay, so the binding patterns we assess through binding energy. Okay, and if you see, however, I mentioned like a receptor here. Here you can able to see the surface structure of the receptor. In 3D structure of a protein, we can see like a secondary structure, ribbon, wire model, etc. Here it we are showing. I just shown you a surface model. So in the surface model, wherever you can see the pouches, wherever you can see the pouches on the surface, we call this as hydrophilic binding sites. And if you find any kind of pouches buried inside, so that if ligand is docked, if you can't able to see the ligand by turning in all the directions, it means that our ligand is docked in the hydrophobic binding site. But see, after docking, the ligand is visible on the surface. So this is our ligand is docking in the hydrophilic binding site. So even through after the docking, we can able to classify the ligand as a hydrophilic ligand or a hydrophobic ligand. Generally, the hydrophobic binds in the hydrophobic binding sites. Okay. Now, let me go ahead with the protein ligand docking. So, in protein ligand docking, we have plenty of tools to dock. But in I believe in 2000, I've used these kind of tools in Anna University, UCSF dock. It's not user friendly. We have to use a Silicon graphics interface, and we have to use only the Unix operating systems. So in Unix only we have to do. But now we can find so many commercial tools developed in Windows. Okay, and you can see almost these docking tools are available in any operating systems. Okay, these are few of the tools. And for protein docking, 3D Docksuit, Bigger, Class Pro, Dock, Etcher. But the popular protein ligand docking tools, at present the Indian scientists are using Autodock. The Autodock, you can find the versions like Autodock 4.0, Autodock 4.0, Covalent Autodock, and we have Autodock PYRX. So just now we have seen one more thing, Autodock Vina. So like this free, exactly the Autodock is not a freeware. It We can work through 15 or 30 days trial version. I believe it is 15 days trial version. After 15 days, we are skipping the license and we are using that kind of Autodock. But few versions we should procure to work with this Autodock. but it's very less expensive when compared to the packages like Discovery Studio. BioVia Discovery Studio, to dock a single ligand with the receptor, we use a module called ligand fit. And if you want to assess the score, we are using a module called ligand score. Ligand fit is to dock a ligand with a receptor. I think you also know the mechanism, like the ligand and receptor. The ligand of particular concentration reacts with the receptor of particular concentration, and a reversible reaction, it can form the ligand receptor complex. And if this is your forward reaction rate constant, this is the backward reaction rate constant. So that is the change of pH can reverse the dissemination of ligand and receptor, dissociate into ligand and receptor from the ligand receptor complex. Okay. So, okay, in Discovery Studio, ligand fit is for talking single ligand and single receptor. And ligand score, we generate around 8 different scores. In Autodoc, we get only one score called binding energy score. But here we can get 8 different scores. And like Discovery Studio, we also have Scroidinger. This is also a popular tool used by many of the reputed institutes. In Scroidinger, we have a single ligand. If you want to dock a single ligand with a single receptor, we can use Surflex dock. And we can use gold for protein-protein docking. Okay, similarly, even in Discovery Studio, if you want to dock a protein-protein, we also have a similar kind of a protein docking server in Discovery Studio. Okay, that is one of the module. Like this, if you want to dock multiple ligands, if you want to dock multiple ligands with a receptor, we use a module called LibDock. LibDock, it means that library dock. Library of molecules you're docking at the same time. Instead of docking single molecule, if you have 10 ligands, instead of docking 10 different times with the 10 different ligands, we can use a protocol, same protocol, and that automatically dock all the 10 ligands with the receptor. That's what we call it as virtual screening. The virtual screening in Discovery Studio can happen through LibDock. Okay, so like this, you can see tripose Cybele. There is one more tool called the Cybele. This is also popular kind of tools. First, I explained you Autodoc, Discovery Studio. Third is Croidinger, and the fourth I am mentioning about Cybele. Okay, and then we have Yasara. Very few are using Yasser, but Scroid in the Discovery Studio or eminent can find in many of the institutions. I believe in VIT, IITs, they have either Discovery Studio or Scroid. Okay, now let me go ahead with this next kind of thing, that is protein-protein docking. So mostly the protein-protein docking, we can able to work with online tools. Free tools are available. So we have to go to that website and we have to upload the two different protein molecules. So examples provided are Haddock, Hex, Azure, Dot, ClassPro. So these are some of the examples. And for docking, as I mentioned, we have freeware tools as well as commercial tools. If you see, this is a freeware tool. This is also freeware, just because we are skipping the license and working. But Affinity, we have to pay. Flex, Biosolve it. So now this is also getting popular. And I think even DNA Star, that is one more kind of a tool called DNA Star package. There also we can able to talk. So these are commercial packages. Okay, Affinity, Flex, Gold, Fred, FRED, and Glide, Scroidinger Glide. In Scroidinger we have Glide. Okay, and then I think I mentioned wrong. Whatever I mentioned earlier, uh, Scroidinger, we are not using Surflex, we are using Glide. In Cybele only, we are using Surflex Dock. Okay. So in Scroidinger Glide for ligand receptor docking. And Cybele only we have Surflex Dock for ligand receptor docking. So a small change, kindly note this correct information. Now this is something about, and of course can see it's available in Science Scroll. Okay, talking in Docker. So talking can also be defined in another way. Like how can be defined? It is the identification of the low energy binding modes. So obviously, if you see in Autodoc, so whatever negative maximum energy we get, that we take it as a best talk pose. And if you see that, if you are trying with a binding site of the receptor, okay, then it is more appropriate. So a ligand can talk in any binding sites of the receptor, but if it is an active site here I mean, which can give an agonism effect, a beneficial effect, a biological effect, a desired biological effect. Okay, so if you have 10 binding sites and if only the one particular binding site is going to give you the desired biological effect, then that is what here they're mentioning like an active site. But generally, the active site should be described only to an enzyme, and the remaining things are all binding sites for a receptor. Okay, now let me go ahead with the steps in talking. We have major steps like ligand preparation, site characterization, that is determining the sites, and the third is the scoring rate calculation, and fourth is the talking protocol. Prediction of the binding energy of the ligand receptor. I already mentioned you how we go with the kinetics. But what is the major thermodynamics, So whatever energy we calculate is not the potential energy, mechanical energy, electrical energy, but whatever energy I mentioned, it is change in Gibbs-free energy. The change in Gibbs-free energy is calculated as change in enthalpy to the difference of product of temperature and change in entropy. So this is how whatever the binding energy in Autodoc in kilocalorie per mole is nothing but change in Gibbs-free energy. Okay, and if you see this del G can also be calculated before the reaction. That is, if you see the del G naught is before the reaction. Okay, before ligand is reacting with the receptor, and if you see the del G naught minus. Okay, del G naught is the free energy associated with the reaction under standard conditions, and R is universal gas constant, T is temperature, and Kd is the dissociation constant. So what is Kd? Actually here I have shown you the ligand and receptor is going to interact to form ligand receptor. So whatever I mentioned is an association. So this is Ka, and whatever is going to dissociate is this is what we can find. The reverse reaction we can find dissociation. So this is Kd. Then how the Kd can be defined? Kd is defined as the product. What is the product? Ligand concentration multiplied by the receptor concentration, and to the ligand receptor complex concentration. See, we'll be taking a particular ligand and particular receptor concentration, and we find the ligand receptor complex. For dissociation constant, we calculate through this formula. And obviously, this is nothing but an inverse of Ka. Even if you calculate Ka, Kd can be calculated as inverse of Kd. Okay, so this is how we can understand the thermodynamics. So how to link the kinetics thermodynamics? Okay, so I'm showing you. So this can be used to calculate the change in Gibbs free energy. And now about molecular talking. So till now we discussed about the kinetics, thermodynamics of molecular talking. Oh, where exactly we find in biological applications, you know, wherever we find the molecular recognition, how we can recognize the molecule. Like how you did you hear something about sensible heat? What is sensible heat? If and if you touch, you feel the kind of heat. That's what sensible. So if only if a molecule is binding with another molecule, then only you can recognize a molecule as bound or not. Then you find some biological effect. Some mechanism is going to happen. See, in this case, I've given four examples. This antigen antibody, okay, where exactly we can find this, we can find in immunology, isn't it? This is a branch of science. And if you see here, the substrate and enzyme, where exactly we can see in enzymology. So in all the biotechnology areas, obviously we find this molecular recognition. I'm giving you the areas of biology like immunology, enzymology, transport and molecule. Can someone give me an example for transport and molecule? I think you know the various examples for the antigen antibody, enzyme substrate. The popular thing is TMB substrate with the enzymes, isn't it? Or superoxidase enzyme, which we find in ELISA. In immunology, plenty of the thing. If you see IL-4 ligand bind with the IL-4 receptor, GMCSF ligand bind with the GMCSF receptor. Like this, you see, whatever 18 interleukins we have, the interleukins have the ligand as well as a receptor. So other than this, even you can see cluster of differentiation molecules also interact with the few receptors. Of course, these are soluble cytokines. Interleukins are soluble cytokines. Even growth factors also have a ligand and receptor. Tumor necrosis factor, interferons, this also have ligand and receptor. So most of the immune proteins follow this antigen antibody mechanism. If you want to develop therapeutics for cancer, the reliable therapeutic for the final stage or just before the final stage is using the antibodies to just increase the life, okay, shell life. And if you see this, the this third example, transport and molecule. Any examples? Can I mention you? So the example what we can take is the oxygen carrying by hemoglobin. The hemoglobin function is to transport gases. What are the gases? The one gas is oxygen, the other is CO2. So during inhalation, oxygen is taken in, and instead of hemoglobin, it is converted as oxyhemoglobin. And during exhalation, carbon dioxide is released. So carboxyhemoglobin protein is formed at that time. So hemoglobin is different, oxyhemoglobin, carboxyhemoglobin is also different. So one thing you have to remember, so if the gas molecule is carried by the hemoglobin, okay, we are studying the transport function. Similarly, we have plenty of examples for the transport and signal receptor. This is most important. Whatever I mentioned as an immunology, the same thing we can also use it here. But the well-known is acetylcholine. Did you heard about the acetylcholine? Acetylcholine is the chemical. Where is it going to secrete? Acetylcholine is going to bind with what kind of a receptors? Acetylcholine. It's a chemical, isn't it? So I'm not making you to secrete acetylcholine, I believe. If I make you to secrete acetylcholine, you can tell me where exactly acetylcholine is going to secrete. Can I get answers from your side? Blood. Okay. Okay. Excretion, kidney. I don't know. Somewhere I missed. Okay. So, you're giving the example for my question, isn't it? So actually, the acetylcholine is secreted in brain, in neural cells. Yes, in neural cells only we find this acetylcholine, isn't it? So if you're thinking it too much, okay, you mean that we are secreting more chemical molecules or binding in our neural molecules, and so we are thinking. I mean, we are getting good memory like that. So okay, now let me go ahead. So you can able to see this molecular recognition in transcription, translation, signal transduction, transport, regulation, enzymatic analysis, viral and bacterial infection and response. Wherever we find the molecular recognition, we find the molecular docking in computational part. Okay, so we can do everything through the computational softwares. Agonist. So actually, the molecular recognition can be understood through agonist and antagonist. So in the molecular docking, one can simply check the binding of ligand and receptor. We can't determine whether is it an agonist or antagonist. Only we can check this binding through binding energy. That's it. So whether the inhibitor or drug, when you can tell it is a drug, if it gives an agonism, if it gives an agonism. The inhibitor can also be an antagonist, can be also be an antagonism. So which is not a desired biological effect. Yesterday we have seen adverse drug reactions, isn't it? Drug reaction. So one thing you have to remember, only the affinity alone we can check. So increase or decrease in recognition is also somewhat difficult to understand. If we see the molecular docking methods, the theoretical methods or Fisher lock and key proposed in 1890, Daniel Koshland in a proposed induced fit in 1958, Boyong Ma, this is the latest thing, whatever we find in from Autodoc to commercial tools, the software uses conformational ensemble model. So that was proposed by Boyong Ma in 2003. The lock and key, simply if our ligand, okay, is fixed and receptor is fixed. So if you find the correct conformation can bind with the receptor, like how a heart lock and key. So this is my key, this is my lock, and in our case, this is our ligand and this is our receptor. Okay, so key lock, the key is small, lock is large. So the whatever key I mentioned is a ligand, and whatever lock I mentioned is the receptor. So fixed ligand, fixed receptor. So here in this induced fit theory, the second theory, you find a small change. So before talking, you find there is a ligand and receptor has a different dimensions, but to just fit, you can able to see both the ligand and receptor or flexible with each other to bind with each other. So this is actually a induced fit where you can see both ligand and receptor are in flexible state. So the third one is the conformational ensemble. Obviously, we follow the earlier model, induced fit theory, in the conformational ensemble. Other than that, we also check the binding energies. So binding energies in different conformations. If we can able to generate around 10 different poses, 10 different conformations, that is a ligand bind with 10 different poses in the receptor, okay, so the which is going to give a negative maximum, that we call it as global energy. We can get only one global energy and the remaining are local energy. So one global and the remaining nine are local. So we are not bothered about the local energy, we bother only the global energy. That only you project and show at the final. So this is what we call it as an ensemble model. Okay, nowadays we are using the ensemble model to predict the binding energies. And the docking we can classify as a small molecule protein. That's what we are going to do with the autodoc. But we also have other tools like protein online tools, protein nucleic acid. More about the protein docking, etc. We'll be discussing in vaccine design. Okay, there is a model called vaccine design where we try to interact with immune protein with another protein. So that we'll be discussing on about that. Now we'll be discussing only the small molecule protein. Also, one thing you have to remember, DNA protein interaction, small molecule DNA interaction. This also comes under docking. We can try to dock any molecule with any molecule. One is small molecule, another is a large molecule. Even in protein, if antigen is a large molecule, or antibody is a small molecule. So this is how we consider this as a ligand and this as a receptor for docking. Now let me take you to the molecular complementarity in computational docking. So in docking, it is not merely fitting one puzzle with another puzzle. First puzzle is no, we generally in docking softwares, we use the chemical complementarity. This is what we're following. What do you mean by chemical complementarity? See the example here. If this is the ligand, which is having a hydrogen bond acceptor, and in receptor, that is an amino acid of the protein receptor, is going to interact with the hydrogen bond donor. A hydrogen bond donor of a receptor, if it interacts with a hydrogen bond acceptor of a ligand, you can find only one hydrogen bond. You can find only one hydrogen bond is formed. Other than one hydrogen bond, if you see our ligand is going to have an aromatic ring, and even a receptor. We have aromatic amino acids, isn't it? So if aromatic amino acid is interacting with an aromatic drug, obviously can see six interactions if it is a six-member ring. So when compared to one hydrogen bond, here we got one interaction itself, we got six hydrogen bond interactions, which is very strong. So if you have these kind of a strong interactions, we can reduce the dosages. You remember when we discussed on about the acetaminophen, we are taking three doses per day. But the same, if I discussed on treatment of bowel, upset of bowels, so where we are targeting more than 7 meters of small intestine and 1.5 meters of large intestine, around 8.5 meters we are targeting the small drug, for 5 mg drug of loperamide. Okay, so that loperamide can also have kind of hydrogen bond interactions like this. Okay, a single interaction can give. So this can reduce the dosage level. That we can understand if an interaction is more. Similarly, the electrostatic, the positive and negative attractions. So positive and negative attractions can give some two bonds. Hydrophobic attractions can have nearly four bonds. So chemical complementarity. This is something we use in the tools to determine the binding energy. And as I mentioned earlier, in conformational ensemble, if my ligand is perfectly binding with the receptor, see the other process, the ligand is not binding perfectly. It is binding somewhere at the top. It was some binding somewhere at the side, so that the binding energy is less. If we perfectly bind at the binding site, then you can find this as a global energy. This is what we report as a kilocalorie per mole. Of course, we use the change in Gibbs free energy formula to calculate. So the types of docking, the only one type of docking which is not possible is having a flexible protein and rigid ligand. You know, the initially whatever I told, Fisher lock and key model uses rigid ligand and rigid protein to dock. Okay, but most of the tools whatever we can see at present to dock has the flexible protein and flexible ligand, so that you can also follow the conformational ensemble method. Okay. Now about the scoring methods, how the scoring is done, how the Autodoc is giving grinding energy, and how the other tools giving more similar scores. I told you Autodoc only one score, but Discovery Studio ligand score can give 8 scores. So there are different physical chemical properties of the ligand and receptor as well as the ligand receptor complex, isn't it? So the most important properties are the interaction energies between the two molecules. The next is the desolvation and the solvation energies. So more about this we'll be discussing during the grid, how to compute the grid. And the entropic factors that occur upon entropic factors. You remember, to calculate the Gibbs free energy, we are also using the enthalpy and entropy. So this can able to determine the Gibbs free energy binding. And if you see the steps in molecular docking, protein and ligand selection, protein ligand. So first we have to prepare the protein and ligand, and we have to check first identify the binding sites of the receptor. Then if any water molecules are there, we can also remove the water molecules because that can hinder docking our ligands in the hydrophilic binding sites. Okay, so most of the times we remove the water molecules and docking, and then we go with the evaluation of the docking. So today we'll be discussing only how to dock. Up to this alone we'll be discussing. So we do from first step to the last step. The evaluation we'll be discussing on tomorrow. So some examples. So today I'll be circulating these journal papers. Very simple to do, simple to work also. Okay, here in this paper, I worked on two different target receptors called PRP1, PA2Y1. These are the target receptors for cardiovascular diseases. I targeted with a plant compound called Vithaferin A. Of course, this is an UGC funded project, minor project with the Rajalakshmi Engineering College faculty received 5 lakhs to work on this. Of course, I helped them to work on computational part. So here we use two different major drug design techniques. One is molecular docking, the other is MD simulation. These are the major things we have used. So and we have published in this commercial tool. If we are using the system biology approach, we can publish even in the high quality publications. So I also will be teaching you the system biology approach in the subsequent sessions. Then I will be sharing you that kind of research papers. So here in this with a plant compound docked with the target receptor. So these are kind of a 3D interaction images I'm showing from this. And this is in another article where I'm talking a plant compound, very we know. I just talked with multiple compounds and I got one compound showing a best kind of binding energy value with these two important receptors of hepatocellular carcinoma. Here also I use docking and simulation. Okay, so docking I did with Autodoc in both the cases, and MD simulation I worked with GrowMax. GrowMax. More about GrowMax MD simulation we'll be discussing in that particular sessions. Here I am showing you the 2D interaction images. Earlier I showed the 3D interaction images. Here I am showing a compound called pyrivinone B interacting with epidermal growth factor receptor. Okay, and similarly, here you can see the 2D interaction image. So what you can find in the 2D interaction, so this is our ligand. Okay, this is our ligand, and outside whatever you see are the binding site amino acids. And whatever green color you find are the hydrogen bond interactions, which shows the stable interaction. And whatever you see here, these are the hydrogen bond length. These are the hydrogen bond length. Okay, so this is something about thing and the protocol how to dock with a small ligand with a molecule that I'm going to illustrate using Autodoc. So I've already shared you the protocol in the WhatsApp. So the first step is prepare the protein file. We have so many sub steps present, and the second is to prepare the ligand. So after preparing the receptor and ligand, then the third step we follow is preparing the flexible residue file. So where we are also defining the binding sites or active sites. And the fourth step we use is the running autoclave calculation. And the fifth is to prepare the docking parameter file. So we complete up to this on today. Analysis of docking results, we will be discussing it on tomorrow, along with virtual screening. That is docking multiple ligands with receptor. We'll be discussing it on tomorrow. Today we discuss a single ligand, single receptor docking. Can we go ahead with the Autodoc illustration? For Autodoc, one should remember that we have to install four tools. We have to install four tools for Autodoc. So if you see what are the most important tools for Autodoc, okay, so fast. Yeah. So the most important tools to install in the Autodoc are the first is Autodoc Suite 4.2.6. This Autodoc Suite 4.2.6 is compatible for both 32-bit and 64-bit versions. This is a common file for 32 and 64-bit. This is one tool. The second is Chimera. So Chimera, I think if you have a recent laptops, you have 64-bit version, isn't it? So we have to install 64-bit version of Chimera and EduPymol 64-bit version. MGL tools, the 32-bit version also works in 64. So Autodoc Suite and MGL tools generally we use 32-bit version, which can also work with 64. But the remaining Chimera and EDU Python molecular viewer, this we have to install 64-bit version. Okay, so 64-bit versions are available. These are free tools to download. Simply we have to type this kind of thing in Google and we can download these tools. So if you download and install all these four tools in random, even you can install anything in random, then you can find the four icons after installation. The four icons what you can find in Autodoc is number one, Python molecular viewer, PMV 1.5.6, CAD T 1.5.6, Next, we can able to find that vision 1.5.6. This is the third icon. And the fourth icon we can see is this Autodoc. This is what we use to open a small ligand binding in the receptor. So after installation of all the four softwares, you can see that four icons in our desktop. Okay. So very small files. It's easier to download and install on the same day. And whatever I work, if you practice on the same day, you won't forget, or else you'll be forgetting how to practice. And whatever videos I share, you slowly watch bit by bit, second by each and every second you play, you pause and work. And however I worked, if you try like that, you will be getting the result. If you make any small mistake, you won't get the result. Okay, and if you see here, I've already installed this tool some 4 years before. So uh, it's asking me to register, but I'll be using Remind Me Later. If I click Remind Me Later, then you'll be getting this tool. You can also able to use the free version of the tool, whatever they provided. So first, to work with this Autodoc, we can have one folder in desktop. So in the folder, I kept a folder called Demo 6. So in the Demo 6 folder, I'm going to keep the four icons, I mean the four files. The first file I kept is autodoc4.exe. After installing that MGL tools from scripts.edu website, we can find this Autodoc 4 and AutoCreate 4 in program files. In program files of MGL tools, you can find these two exe files, executable files. So for conveniently using that, I just copied from the program files and I pasted in the desktop folder. Okay, this is a pre-preparation. And then I'm also copying and pasting my Autodoc tutorial. And presently, I just downloaded one target receptor called protease. So this protease is a small molecule. So for illustration, I'm going to try with this small molecule talking so that I can able to complete within the stipulated time. So, but I didn't download the ligand. If we want to download a ligand, as usual, what we can use NCBA PubChem. Okay, use NCBA PubChem or DrugBank. PubChem is easier for us. Can we try with curcumin? A kind of plant compound. So today I'll be trying a talking curcumin with the protease. So for downloading the curcumin, I go to the curcumin, okay, and I can download the 3D format. So only 3D format you have to download. Don't download the 2D format. And 3D format, I prefer to use the STF. I going to click the STF, that is structured data format. I'm going to save it in demo folder. So whatever I'm saving is curcumin, okay, which is in STF format. But if I want to have in different format, I can open in Discovery Studio Visualizer and I can save in different format, whatever I require. At present, no need. Let me take you to the tutorial. So this is my tutorial. So what is the first step I have to make? So if you see here, this is the icon for the Autodoc. When I double click and open the blue color icon, the first step which I didn't mention in that tutorial is first set the path. This alone I didn't mention. The remaining steps I have mentioned. The first step I suggest you to go with file preferences set. In file preferences set, you check the folder, and the folder I provided is demo 6. So I just copy and paste it in the startup directory. Then click make default set. Okay, in startup directory alone, we have to paste the URL of the folder. So URL of the folder is just have to click and copy, and then you have to paste it here. Already done. So make default set I have completed just now. Dismiss. Now let me go ahead with the first step. The first step is file read molecule select protein name. File read molecule. The selected protein is protease. Okay, I uploaded the protein. The first step is completed. Adding hydrogens. Edit hydrogen, add all hydrogens, and then add Coleman charges. First edit hydrogens, add, default all hydrogens is present. Simply click OK. So after adding hydrogens, you can see a small conformation changes happened. The white color thing, whatever is your hydrogens added on the amino acid surfaces. The next is adding the charges. So add Coleman charges. Charges, add Coleman charges. We got the Coleman charge. Simply click OK. So this step is completed. The next, the final sub step in step 1 is file, save, write PDB. OK. File, save, write PDB. Okay, here in this folder we have protease.pdb. I'm going to rename as protein1.pdb. Protein1.pdb. Okay, this is how we are going to rename. Click OK. Now this is completed. The first step is fully completed. I have saved up to protein1.pdb. So I prepared the protein file. The next is to prepare the ligand. Ligand input to open ligand. So ligand input open ligand. So in this we don't find the ligand because we have saved the ligand defaultly in STF format, structured data format. So we have to click all files to see the ligand. Yes, our ligand is visible. So open this curcumin. So unknown ligand. See, STF is not accepting. If STF is not accepting, then we have to open in Discovery Studio Visualizer and we have to save it as like .pdb. So STF format is not supported by Autodoc. So I'm going to open this in Visualizer. As I mentioned on yesterday, whatever curcumin structure you find here, okay, that you are going to save as, okay. In Discovery Studio, you have something called PDB. So select that PDB and save this curcumin.pdb. Now I have saved. Can I open this now? So in that step, uh, so ligand input open ligand. Ligand input open ligand. So PDB instead of PDBQT, click all files, go with the curcumin.pdb. Yes, curcumin is opened, but see somewhere you can find here. Okay, we are not yet talked. Simply we just opened it in the visualizer. The next is ligand torsion tree detect route. Ligand torsion tree detect route. The next is ligand output save. Ligand output save as PDBQT. So I am going to save as ligand1.pdbqt. So now I converted the ligand as PDBQT file. So that's the accepted format of Autodoc, any Autodoc. And the next step is third step, grid macromolecule choose. Grid macromolecule. So wherever I mention like choose, better use open option. It will be easier for us to understand. So instead of choose, go with open. Grid macromolecule open. So macromolecule is not shown. Only the ligand or PDBQT is shown. What is the reason? Recently, whatever I saved is protein1.pdb. So we have to save the protein1.pdb as protein1.pdbqt. So click all files. Check where is our recent protein. This is our recent protein, which is in PDB file. So just click open. If you click open, it prompts you with this kind of a box. Simply click OK. Now it is asking you. Now it's asking you defaultly to save it as PDBQT format. I am saving it as protein1.pdbqt. Click save. Okay, so we have completed a part of the third step. So in this part, up to this step, we have saved protein as PDBQT and here ligand as PDBQT. So preparation of ligand and receptor is completed. The next is to define binding sites. For this, select from string. Here in molecular list, we have to open only the macromolecule. The recent macromolecule out I saved is protein 1, isn't it? So click this protein 1. And in atom, I'm going to type asterisk. Why I'm selecting asterisk, if you want to select all the atoms in a protein, like it can be, the binding site can be anywhere in this protein. So I have to select everything. So I'm simply have to type the asterisk in the atom. And remember, this is the most important step. We have to click only once add. If you click once, you can see this yellow color. If you click two times add, then obviously you have to close the window and you have to start from the beginning. You have to start downloading the files from beginning and you have to make it, because you won't get an output if you make any wrong. So however I mentioned, however I you follow the step. Later click dismiss. Okay. So now we have to going to define the binding sites in anywhere of the routine. Now the fourth is running autogrid calculation. Grid set map types and go with the open ligand. Grid set map types open ligand. Even though I mentioned like choose ligand, I told you better go with open option, which will be easier for you. So grid set map types open ligand. Here in these two formats, which is ligand, ligand1.pdbqt. This is the ligand. So select this ligand, click open. So automatically something is added and the ligand is done. So this step is completed. The next is grid box. Grid grid box. So in this grid box, you can see a cubical box. One. Now we can see a cubical box. See, if only if this cubical grid box is covering the entire protein, then only if you fill water in the grid box, you can see the water can be accumulating in the outside surface of the protein. If water is present on the outer surface of the protein, we call that as hydrophilic binding sites. And if instead the buried side of the protein, you can't, you, is it visible, participants, whatever I'm showing you in the Autodoc? Yes or no?
Yes, sir. Yes, sir.
Ah, yeah, yeah. Okay. So, actually, if the water molecules are present on the surface, we call that as an hydrophilic binding sites, and if water is not getting inside, that is hydrophobic binding sites. Now I simply increase the dimensions of the grid. So if we want to increase the dimensions, I right click this, right click apply. Now you can see everything is getting activated. I'm going to increase the size of the value so that even the maximum will apply. Then click apply. So I increase the size of the grid box. I have to enclose the entire protein in the grid, or else because you will be missing the binding sites. The ligand is not going to bind in the anywhere of the binding site. So I just increase the size. So I am increasing the dimensions in all the three directions. So click apply, everything get activated. Simply increase the size. So now if you see this, my entire protein, the protease is fit in this grid box. Okay. So after making this, file close, saving current. This is the step we have to make. File close, saving current. So we have enclosed in the grid box. So up to this is completed. The next is grid output save as GPF. Grid output save as GPF. So we'll be saving it as grid1.gpf. GPF stands for grid parameter file. We are saving this as grid1.grid parameter file. Later, run autogrid. Run autogrid. So here in autogrid 4, you have to click browse, select the autogrid 4 exe file so that the entire URL is visible. Now grid1.gpf, you select this browse, the next immediate thing, select this grid1.gpf. Even you got another thing automatically added, grid1.glg. GLG stand for grid log file, grid parameter file, grid log file. So also this is up and down. Now simply click launch. If I click launch, you'll be getting this white window. If this white window immediately disappears, then we have made some mistakes in the protocol. If any of the step is wrong, then this white window immediately vanishes. But it is staying for some time, isn't it? Because grid calculation is running at present. And based on the size of the protein and size of the ligand, whatever we are talking, you can see that the white window stay up to that. If I use a large molecule, okay, so see immediately disappear because both the molecules are smaller. If both the molecules are larger, it takes more time. Autodock can accept around 500 molecular weight of ligands to dock. Now let me go ahead with the next protocol. Preparing the docking parameter file. Docking macromolecule set rigid file. Docking macromolecule set rigid file name. So here we are asking to open the macromolecule. The macromolecule is protein 1.pdb. Simply click open. So after opening this, docking ligand open. Docking ligand, I mentioned like choose, better instead of choose, get use this option open. So we are asking to open the ligand. So click this ligand 1.pdb open. Yes, we are getting this kind of a window. Simply click accept, which is default thing. So we have completed up to this. Only three steps are left out on today. One is docking search parameter genetic algorithm. Docking search parameter genetic algorithm. So this is the next step. We are using genetic algorithm for docking. Here we are going to change the genetic algorithm runs as 2. Instead of getting 10 different conformers of ligand docked with a receptor, if we get a best 2 conformers, that is sufficient. So in our publication, we need only one result. Even we don't need 2 results. If we want to get best 2 results, we can show the 2. Why we have to take 10 results? There is no need to show all the 10 results. So I simply reducing the time, or I'm reducing the number of genetic algorithm runs. Simply click accept. So this step is completed. The next is docking output. Oh, this I did, I believe. No, what I'm doing. So doc1.tpf. Okay. So I'm here one day I'm saving it as doc parameter file. So that is after doing this genetic algorithm, I'm completing this step. Docking output American genetic algorithm. And then run autodoc launch. Run autodoc. And here we have to select autodoc 4. Click open and browse. Doc1.dpf is the default thing which can open. So click open that. Whatever we get in default, we have to select and we open. So you find all these URLs at present. Simply click launch. Again, a white window can appear. But it will take less time to close when compared to running the grid. So this is the step we are making our ligand docking with our receptor. So this is coming under uh, the fifth step. After this fifth step, in sixth step, we are going to save this ligand receptor in the form of complex file. Okay, so still it's showing. If that white window closes, then we can execute this sixth step and we can close the window. So the analysis of results, I showed you how to analyze using Python molecular viewer. But I'm going to provide demo with Discovery Studio Visualizer on tomorrow. Analyzing results, we'll be discussing it on tomorrow. Today will be completing up to docking and saving the ligand receptor file in the form of PDB format. Okay, that's automatically closed. So I'm going to with the sixth step. Analyze dockings open. Analyze dockings open. So here I'm going to open the default thing doc1.dlg. So whatever you get in default, click OK. Again OK. So this step is completed. Next, analyze macromolecule choose. Instead of choose, open. Nothing happened. Something has happened. No need to worry. It's already done. Analyze conformations load. Analyze conformations load. So we got the best torque energy is minus 6.18 kilocalorie per mole. This is the best first torque conformer. The second torque conformer is minus 5.39, which is insignificant. In Autodoc, whatever scores minus 6 and below are significant scores. Fortunately, for the first course, we got minus 6 below, minus 6.18. Not only we get the binding energy, we also get the Ki values. So also we can show this as an as an output. So we have completed up to this. Analyze conformations, play ranked by energy. So after this, click this ampersand. Now it's asking you to build current. Whatever ligand and receptor is bounded, we click build current, and then we click write complex. During the write complex, we have to select all. And now what is our ligand? Curcumin and the protease. Okay, dot pdb. Whatever I am saving like curcumin dot underscore protease dot pdb, it will be saved like pdb format. So this is a complex file, curcumin docked with a protease. Simply click save. So if and if you keep it in double quotes, it will be saved like .pdb or it will be saved with some other extensions. If you have completed up to this step, you can close it, close the window, and you can analyze the result on any day at any time. Okay. So up to this, we have to complete. Then, or else if you close in between, we have to do step from beginning. If you do two steps up to 4 steps, then again we have to download the files and we have to make the steps once again. See, these are the intermediate files formed after the docking. So analysis of the results we'll be discussing on tomorrow with the intermediate files. Okay. So today I have completed session on how to go with docking using Autodock. What is the range of binding energy, sir, in kilocalorie per mole? Actually, -6 and below, like few ligands and receptors, if it has more interactions, it can come around even -14, something like that. So even the lowest score, you can get up to -3, -4, -5 also. Okay, so this is what I think I have done more than 2000 dockings. This is the range I found in these dockings. I mean, how can we come to know which is best based on binding energy? And does negative score also can? Yes, negative score is what the preferred in Autodock. If you get a positive score, that is not docked perfectly. So the energy, if you know that in the universe, entropy always tends to increases, and similarly, Gibbs energy always tends to decreases. If I'm taking the class, the Gibbs energy is going to decreases. Similarly, if you're hearing, Gibbs energy decreases. So the negative energy is what preferred, even in ligand receptor binding. Any other doubts? I completed the session. I think I also cleared his query. Any other doubts you can ask me. So I thank you once again for your patience and interaction from the beginning to the end. So we join the next session that's on analyzing the binding and also to work on virtual screening using Autodock PVRX on tomorrow. Tomorrow also we have similar kind of hands-on session. Don't miss to attend. So thank you. Good night to everyone.